Good day and I welcome you back to our channel. Today we're going to discuss bacteriophages, and this one is part one of the two-part series where we will use discuss about bacteriophages. Now, wetin bacteriophages be? Bacteriophages na special kind of viruses wey get the ability to invade bacteria. They enter into bacteria, use up everything wey bacteria get for im body, and they go use am form newer bacteriophage. Now, bacteriophage dey very popular because they dey very present in nature, and then they help to shape bacterial population because they feed on a lot of bacteria. So bacteriophages dey very popular. I don use one word before, ubiquitous, and this word bin mean say dem dey present in every environment, every different place, for water, for land, for sea, everywhere where bacteria dey, bacteriophages dey, and this one dey important. Now, the structure of bacteriophage too, e dey important as a science student to understand wetin the structure of bacteriophage be like. The structure of bacteriophage, you get four major elements, four major structure wey dey for inside bacteriophage, for which we go fit understand say okay, this thing wey we dey look at na bacteriophage, na virus wey dey invade bacteria. Now, these four structures na: one, we get wetin we dey call capsid or protein coat. That one dey. Second one wey dey there na viral genome. Viral genome, that one fit be DNA or RNA. Then the next one na wetin we dey call tail pins or tail fibers. And the fourth and last but not the least na wetin we dey call base plates. Now, make we discuss these four structures one by one. First, capsid. Capsid, we don learn capsid, we don do video on viruses before where we been discuss about capsids. So capsid dey, dem be protein coats. They are formed by wetin we dey call protomeres, and they dey surround all these bacteriophages. They fit get many different kinds of shapes. E get shape wey we dey call polyhedron shape, e get shape wey we dey call icosahedral. I don talk this icosahedral before, say na shape wey be say you go get 20 equilateral triangles, their faces, with 12 vertices or 12 angles. So this one dey important to know. These na some of the shapes wey capsid dey get, and this capsid na protein coat wey dey surround the bacteriophage. After capsid, the next structure na the viral genome. We know say genome na genetic material, so that genetic material fit be DNA or e go be RNA, and e fit be wetin we dey call single-stranded DNA or double-stranded DNA, or e fit be single-stranded RNA or double-stranded RNA. All these different kinds of genetic material bin dey, and na wetin we dey see for inside our bacteriophage. Now, the third one na the tail pins or tail fibers. This one's too dey important. If you see structure of bacteriophage, you will see those things wey be like tails or be like pins wey dey comot for the body of the bacteriophage. And wetin these pins dey do? These pins dey help the bacteriophage attach to the bacteria. The bacteria get wetin we dey call receptor sites. Na places wey be say the virus fit attach itself to. So these tail pins, they dey sabi where these receptor sites dey, and they will go attach themselves to the bacteria through their receptor sites. So dem dey very important. Now, the last one or the last structure wey dey for our bacteriophages na our base plates. These base plates dey the body of the bacteriophage, and they also dey important because when the bacteriophage wan attack or attach itself to the bacteria, na through these base plates the bacteriophage fit get closer to the bacteria and inject the material wey dey for the bacteriophage. So this one dey important for us to know. Also, e dey important for us to understand say bacteriophage, remember say they be viruses, so they get life cycle, and their life cycle dey very similar to the life cycle of normal viruses wey no be bacteriophages. So I will take the steps of the life cycle of the bacteriophage. The steps dey start from: one, attachment; two, penetration; three, na replication and transcription; and then we get assembly, and we get maturation and lysis. These na the stages of the life cycle of the bacteriophage. Now, make we take am one by one, discuss all these different stages. The first one na attachment. These phages, you know say phages na another name wey we dey call bacteriophages. These phages dey go attach themselves to the bacteria. And remember, I tell you say they go attach themselves using their tail pins or tail fibers. So once they do this attachment, na wetin we dey call, once they do this attachment, that one na the first step, attachment. The second thing na penetration. Once these tail pins don attach to the bacteria, the base plate go come closer, come inject, the bacteriophage go come inject in viral genome, whether na DNA or na RNA, and then whether na single-stranded or double-stranded, e go inject that thing into the bacterial cell. Now, that point na penetration, we dey call that point. Once you don inject am, the next thing na replication and transcription. And wetin dey happen for here na say this viral DNA or RNA wey don dey inside the body of the virus, of the bacteria, go come begin dey replicate, dey make many copies of itself. And wetin e go use make those copies na all the genetic material wey the bacteria bin carry. So this bacteriophage wey be virus don attach himself to bacteria, don enter inside bacteria. E go come kolobi all the genetic material wey dey for the bacteria, wey be the bacteria own. E go come take am, come use am forcefully make many copies of itself. So this one na replication or transcription. Now after this one don occur, the next step na assembly. And for assembly, wetin go happen na say new capsids go dey formed around all these new viral genomes wey we don form. Viral genomes wey we don form using the bacteria own DNA. So around all these small viral genomes, capsid, we go create capsid all around them. And when this one don occur, the next thing na maturation. This new bacteriophages now, wey don plenty, wey dey inside the bacteria, they go come mature. Once they mature, the last stage go enter, wey be lysis. They go break through that bacteria, because that bacteria now no fit sustain life again. Everything wey e get, the virus don come replicate inside it, use all its own nutrients, use all its material, make new viruses. E go come burst the bacteria, and all these new viruses go come comot from the body of the bacteria. This na how e dey happen. So that brings us to the end of our discourse on bacteriophage. Thank you. Part 2 still dey come. So keep yourself peeled and your eyes peeled. Please like our content, share our content, and have a wonderful day.